Good morning, good afternoon, and good evening. The room here is pretty full. Mr. Mike Say just arrived, which is great to have our friends from Genome Prairie in the room. Hey, Mike. And we have over 90 registered participants online. Not sure how many are on right now, but again, it represents a global audience. I want to introduce myself. I'm Steve Webb. I'm the CEO of the Global Institute for Food Security at the University of Saskatchewan, or GIFs, and I'll be emceeing this morning's event. We're excited to connect with you all here in Saskatoon, and I want to acknowledge that we are gathered here today on Treaty 6 territory and the homeland of the Métis. We pay our respects to the First Nation ancestors and Métis ancestors of this place and reaffirm our relationship with one another. And I was talking to Gord to tell him to sit down, but there's an opportunity that Gord and I are going to be meeting to talk about how do we drive training opportunities. And again, thanks to Mike and the team at Genome Prairie and Genome Canada for looking at and creating opportunities to create inclusive and growing the training opportunities here in Canada. Today, we have an engaging program for you, and we're looking forward to a great discussion. Again, we'll have microphones because we want the people online to be able to hear your questions and the answers. And we'll be taking questions from the people that type into the chat. We want to take a few moments also to celebrate a recent accomplishment for our team here at GIFs. Earlier this year, GIFs was announced as Canada's first Revio PacBio Certified Service Provider, placing our expertise and capabilities alongside some of the most renowned labs, service providers in the world. This is a significant accomplishment for our team at GIFs, which is bringing new services and capabilities to researchers and organizations throughout Canada and around the world. Again, this is an opportunity to kind of showcase the quality of work that we do, and there's going to be a lot more on that later in the session. Logistics. To talk about some housekeeping items, again, I want to thank members of the GIFs leadership team that are here, as well as all the other GIFs folks that are in the room, especially our speakers, Austin and Andy, because without you, that's where the fun part of the science comes in, not listening to me. I want to also thank PacBio. I want to thank Grace for coming here, and we'll talk a little bit more about what Grace is going to be doing. I see everybody's got a ticket, which is great, on the tables here. And Turner, who's in Indianapolis, is going to be one of the speakers as well. Now, we're going to proceed through the program without a break. And again, for us in the room, there's a complimentary lunch that follows this session. We'll do a short Q&A after each of the speakers. And again, we'll be looking at taking the questions in the room by the mic so the people online can hear. And the folks online, please type your questions into the chat function. And we'll try to get all the questions answered today. If we can't, we'll reach out to you afterwards. And again, connect with GIFs through Partner With Us at gifs.ca as well. And again, I want to work with everyone here to move this along. Now, this is my part to be the ad at GIFs. So, Scott, I apologize. It is an ad for the Global Institute for Food Security. And we were created in 2012 through a unique partnership between government, industry, and academia. And there's not many of those types of organizations around the world. There's several, but not many. Our mission is to work with partners to discover, develop, and deliver innovative solutions for the production of globally sustainable food. And today, we act as an innovation catalyst and a connector with world-class capabilities, which we're highlighting one of those here today. And we work up and down the entire innovation pipeline. Our ambition is to be the preferred partner, bringing the capability, capacity, and the competency of our people, our most important asset, to help our partners, our customers, and our stakeholders advance their research programs and their work. GIFs is one of only a handful of organizations worldwide that have the capability, capacity, and competency that we have here in Saskatoon. We are a global organization. And it's not about what work do we do around the world. It's about the fact that we punch way above our weight and we attract business to Canada and Saskatoon, which is evidenced by who's online here today and our customer base, which reaches right across the country and around the world. At GIFs, our playground is the ag and food sector, but we play up and down the entire innovation pipeline. We work with researchers at universities, researchers at government institutions around the world. We work with industry. We work with everyone in this ecosystem here in Canada and, as I mentioned, everywhere else. We sit in the middle of the innovation pipeline. We act as that connector and bridge. The scale and capacity we have allows us to help drive innovation at the business center and drive invention at the universities and research institutes here in Canada and around the world. We operate at scale. And one of the things that we do have here, in addition to being PacBio's first Canadian certified provider, we are also PacBio's only Canadian provider that has an automated workflow. So when you come to see the Global Institute for Food Security, there's more and more robots in the labs because we need to be thinking about working at the scale of 100, 1,000, 10,000, 100,000 for some of our partners and stakeholders throughout the system. We were created to be that connector and that catalyst. We do not do research on our own. We only do research with our partners. And our goal is to make our partners and customers successful, because when they are successful, we are successful. At GIFs, we have, again, a structure that's a matrix structure. We have 2 market-facing programs, the FCC Accelerated Breeding Program and biomanufacturing, that are supported by state-of-the-art at-scale technology platforms. And we're going to be talking about 2 of them today, the Opal sequencing facility and the data management and analytics group, because again, the ability to provide sequence data is good, but to be able to convert data into knowledge and information is even more important. And at GIFs, we have both of those. Each of these platforms is unique and cutting edge, and really here in Canada, we're the only one that has this type of structure, and globally there's probably only about 3 around the world that look like us once we bring our engineering biology platform online. Again, our mission is to be the one-stop shop, to be able to act at scale for both all the players in the ecosystem, from individual researchers up to corporations, and to be focused on outcomes and impact, making a difference in the marketplace. And our goal is to be self-sustaining. Our Omics and Precision Ag Lab and our data management groups are the 2 platforms that I mentioned that we'll be talking about today. They're powered by being able to do both short and long read sequencing. And again, we're here to talk about the cutting edge technology that PacBio has provided through the Revio and that end-to-end workflows. And again, we have also got excellent data management and analytics that allows us to, with workflows, to visualize the data. And I had a chance last week to see some of that action in progress for a project that we have with a partner here in the ecosystem in Saskatoon. So we have a commitment at GIFs to be committed to innovation and to continue to invest in being at the cutting and bleeding edge of where these technologies take us. This machine here we brought in 2023. We were excited to add the Revio because in 2022 we were excited to add PacBio Sequel 2. And we ended up upgrading. Why? Because of the demand in the ecosystem, the additional capacity that this machine offers, and the speed at which it can provide the results back. 15x capacity from what we had before. That scale is critical for us in our mission and to be able to service and complement the ecosystem. And now, as a PacBio Certified Service Provider, our customers and our partners can see the value of working with the Global Institute for Food Security. And again, I think this is a real good opportunity for us to be looking at how the technology goes towards making a difference in the innovation pipeline. Research is good because research is the start. That's where inventions happen. But the ability to translate and scale it so that it makes a difference in people's lives is where we really need to keep focused on. And because we can be that connector up and down, we're able to help bring these types of technologies to everyone throughout the ecosystem. Now, I mentioned that Grace is here in the room, and Turner is online. And we're going to invite Turner Duncan, Senior Genomics Specialist at PacBio, to say a few words in a moment. I want to say that Turner is based in Indianapolis, Indiana. I spent 19 years in Westfield, Indiana, and the month of May in the state of Indiana is special because that is the 500. And I, of course, had to ask Turner when he was going to the track. He's going to be at the track on actually the best day to be at the track, which is Carburetor Day, which is the last time they can run the cars before the actual big race. And it is, if you've never been, and even if you're not a racing fan, go see the spectacle that is the 500. Anyway, that's nothing to do with today's talk, but just a gross advertisement for Tourism Indiana. And it is a really cool month. Anyway, as I mentioned, Turner has spent the last 9 years helping researchers unlock insights into the possibility of highly accurate long-read sequencing technology that PacBio offers. And again, please join me in welcoming Turner Duncan to say a few words.
Hey there, everybody. Thank you very much for the great introduction, Steve. That's where I can confirm I do have my Indy 500 tickets. So again, thanks for making the plug there. Got them right here. They're ready to go. But yeah, I guess I'll go ahead and share my screen, and that's where, I've got a couple of really exciting things that, we're really honored to be able to spend the time to, share with, everyone at GIFs and your community's ecosystem. So thank you very much. Let me know when you guys can see my screen.
We can see it.
Okay, awesome. So then I guess with that, I'll go ahead and kind of, get up and going. So I guess that's what kind of with the time that, we have today, that's where, our goal is to do a, kind of a PacBio who we are technology overview for anyone who doesn't know who PacBio is, what we do. And then I was going to go into a review of our applications, primarily focused on, whole genome sequencing, full length RNA sequencing, and then microbial and metagenomic sequencing. And then I was going to go into a systems and project design considerations. And so I know that I think, prior to this event, the group here has kind of been made aware that PacBio is doing a Kinnex RNA sequencing type of a promotion and offering. And so I guess pay attention throughout this because we're going to talk about some of that. And I know Steve, that's something that we'll kind of cover here at the end. Is that right?
Yes. Yep.
Awesome. I guess, is there anything else that you'd like to make sure we take care of before I kind of start getting into some of the, the scientific stuff here?
I think the people want to hear the science stuff more than they
Sounds
wanted
good.
to hear me talk about stuff.
Awesome. Yeah, we'll go ahead and dig in. Thank you guys again very much for having us here to talk. So if you're not familiar with PacBio, that's what, our mission is to enable the promise of genomics to better human health. And we're doing that by creating the world's most advanced sequencing technologies. As Steve mentioned, GIFs has had multiple iterations of the PacBio sequencing platforms. If PacBio is not a company or technology you've kept up with, there are a couple of key things that I'd really like to update you on. The first is the advent of our HiFi, or what we call our high fidelity sequencing data type. This was first, kind of published and made possible in 2018. This is what allows PacBio's data to be both incredibly accurate and a long read sequencing technology. When I say long read sequencing, many sequencing types from short reads, primarily Illumina sequencing, are typically in the 1 to 300 base pair range. PacBio, we typically, look for that, 500 base pair to up to 25,000 bases or 25 KB. So our HiFi data type is, when we first had an accurate and long sequence, that differentiates us from both the short read and other long read sequencing technologies. Kind of move forward to the most recent years. That's where we've launched our Revio system that the team there has on site and is Canada's first provider certified service provider for. We also recently launched our Vega benchtop system. The Revio system is our higher throughput, lowest cost per data point system. The Vega is a smaller system that just allows for PacBio technology to be into different groups and different labs. I think we're really excited about the Revio as it's brought the cost of a 3 gigabase size genome down significantly. That's where we recently launched the Spark chemistry that I'll talk a little bit more about here in a bit. But it really does help bring that that cost per data point down. So kind of looking into, what is PacBio sequencing? Why is it unique? How does it work? The key thing that we start with is we start with a double-stranded template. We then put our hairpin library prep adapters on it that circularize this molecule. And then we do our sequencing on a polymerase. So kind of like this little, example you see here, because we have a circularized molecule around a strand displacing polymerase, we're able to sequence a molecule multiple times. And so when we sequence the same molecule multiple times, we can then make, a collapse that down into what we call a consensus read, which is highly accurate. So that's where if we read a molecule one time, we know we're at about, 90% accuracy. If we read it 8 times, we know we're at, Q30 or 1 in 1,000 accuracy, which is similar to your short read or Illumina sequencing data type. So this is kind of, this HiFi data is how we get to both a long and an accurate read. And again, we kind of optimize this for up to 25,000 bases as an insert size. One of the other unique capabilities of this technology is because we don't use PCR anywhere in our library prep or our sequencing systems, we're sequencing native molecules of DNA. And so this allows us to also detect different types of methylation information. And the way that I kind of think of this is if our polymerase is, recording our A, G, C, or T as we're sequencing, the methylation data that we're able to get is kind of interpreting the hiccups of these bases being incorporated around certain different methylation motifs. And so what we're able to do is take this hiccup or what we call our kinetics data, and we're able to run that against a model for something like 5MC in the CBG context, or 6MA in an assay we call FiberSeq, and we're able to analyze these little hiccups in the kinetics data and give a methylation call alongside all of our base calls. So with this technology, you're not only getting A, G, C, or T, but you're also able to analyze this kinetics data to find additional omics information, all with just one library and no additional sequencing types. Kind of just a really high level example from the human genome spaces. This is just showing, between two phases. So a maternal and paternal phase from, one individual. We see that, there are differential methylation between the two. So they would be inheriting some from, one parent and one from the other. So this is a really basic example. But this is something that you're able to, kind of see and use with this methylation information that is really kind of unique to this data type. Another thing I want to point out, what you see here is what we call a smart cell. This is, the unit in which we do our sequencing. On this smart cell, on the Revio system, there are 25 million what we call zero mode waveguides or ZMWs. The idea is we get each one of those to capture exactly one of these molecules. And then we do our sequencing, on this, and we have an optical system that's, kind of looking at each one of those 25 million wells and doing the sequencing. And so it's really a incredible amount of, both chemistry, biology, and physics kind of, all into one system. So it really is pretty amazing how it all works together. So I think when we kind of look at what this data type allows, it's something that you can use for a variety of applications. But the key things are it's a long read system, it's highly accurate, it's uniform in coverage. We don't have GC bias because there's no PCR anywhere in our library prep or sequencing. And it allows for native molecules, which allow us to get this, methylation information I described. So when we kind of think about, moving into the application space and how people are using this, one of the most important and largest use cases for this right now is in creating de novo assemblies or assembling a genome when you don't have a reference. And so that's where PacBio, because we are both accurate and a long read technology, has really been the key component of many of the recent telomere to telomere assemblies. So these are, if you're not familiar with them, gold standard assemblies where they're trying to really resolve every base pair, in a sample. And so I think this first started with humans, but they've been expanding this methodology to, other organisms, including, apes and other plant species. And we're kind of seeing the methodology used here, adopted across some of the larger consortia. And that's where the basis of this type of work really is this PacBio HiFi data type. And these are just, again, as I mentioned, a couple of the, of the large consortia groups that have standardized their de novo genome platforms using HiFi as the base of their work. And that's where the reason that, again, we're kind of doing this is because the more complicated you get when you get to, high levels of ploidy that we know are present in many plant and other species, you really need a large, accurate puzzle piece to kind of put these things together. So these are just kind of a couple of examples of ones that, are explaining why PacBio technology is a great tool for this use case. We were really excited last year. There was a group that, published and generated what is likely the most complex reference genome ever assembled. It's a dodecaploid sugarcane. So that's where, 12 copies of each chromosome. And that's where what we're looking at, here is the first, kind of draft from a while ago, all the way up through the, some of the other different ones and what we see and how much more information and complexity that we've been able to, kind of assemble and elucidate throughout different iterations of assembly of these sugarcane lines. So outside of just, large and complex species, this technology is really suited for, everything from, the large complex to ones that have never been sequenced before, but also incredibly small species as well. And when I say small, we have groups that have used some of our technologies from, individual, insects. And one of the things that I kind of wanted to share with this group is we have a new technique and protocol that we recently launched called Amplify that allows us to start with, as low as 1 nanogram of input material and allows us to, still sequence, from a single individual. So this is really kind of an enabler for very small species. We also have worked with groups to, standardize and take the HiC application that has typically been done on short reads, and develop a way for that to be used with PacBio sequencing. And we call that kind of like the sci-fi application. This amplify workflow, has also been used with, really low input or degraded things like FFPE or, biobanked or ethanol preserved samples. So this is something where, this really kind of helps enable sequencing of things that have been difficult before with a long read technology. Here's kind of just the publication I mentioned of, a group that we worked with the USDA. I think there's a group out in Hawaii, where they look to take this Amplify protocol plus HiC, and that's where they're able to do it from, an individual, insects, which is pretty remarkable. So moving beyond just, the AGCRT, I mentioned that we do enable methylation from a different variety of motifs. One of the ones that, we have seen great adoption of and are working to help commercialize ourselves is what we call the fiber-seq assay. So what it is a library prep process that allows you to treat your library with a methyltransferase that allows you to get information for chromatin accessibility. And so when you use this type of, a library prep, it allows you to get your A, G, C, and T genome like people already get. You get your CpG methylome, and then you also are able to get, this chromatin accessibility genome as well. So it's just we're starting to see this technology as, one where you can consolidate other technologies into one assay. And that's kind of what's really exciting. And I think we've even seen groups start to use this in plant species, specifically looking at, transposable elements, and kind of seeing how, this accessibility of chromatin, impacts, development and evolution. So it's one where it's pretty much a straight up free and easy thing to add to any data set when you use PacBio. So I think to kind of summarize, our whole genome, application, that's where with a PacBio HiFi genome, you get, the SNPs and indels and CNVs you get with your short read data, but you then add a lot of different types of variation that you can really do with one assay. Where if you were to do or want to record these other types of variation, you need, additional secondary assays. So PacBio is both the most comprehensive, um, and allows you to find things that you can't with other technologies. Kind of moving to a different application type, we're going to talk now about full-length RNA sequencing. I think the important thing to point out is, our sequencing is at that isoform or transcript level of resolution. Lots of short-read RNA sequencing, is looking at what genes are there. PacBio full-length RNA sequencing is looking at which transcript of said gene is there. And why that's important is, most of us are probably familiar with the central dogma of biology, DNA, RNA, protein. What we see is, your DNA makes messenger RNAs. And at least in the human area, we see that the majority of genes have more than one isoform or more than one copy of, an mRNA. On average, there's around 7. And we see that, many of these are associated with, diseases and alternative splicing. And we also know that, the different varieties of isoforms are often invisible to short read sequencing because you're just not sequencing enough of the gene to know, which orientation or full structure it is. So this is kind of, what you get with PacBio sequencing. Here on the right, when we do our RNA sequencing, you should see the full 3 and 5 prime end and just know that the transcript or isoform you're sequencing is full and intact and it's there and it's real. With short read sequencing, you'll get a little bit of that and you'll look to do a mapping approach. But if you don't see these, splice junctions, all you can say is this gene was there. You can't say what orientation that gene is in, which is closer to, the protein and the actual biology of of the samples you're sequencing. So it's a whole nother level of resolution that PacBio RNA sequencing allows. This is just a really high level example of, um, in some customer samples where certain expression of two isoforms increase mineralization, where expression of two separate isoforms can decrease mineralization. So the exact isoform that, is active in your sample can be the difference in the biology that you're seeing downstream. And so if you can't, elucidate this, you might miss some of the mechanisms of action for why you might be seeing things in experiments. This also is something that is incredibly useful and possible with single cell RNA sequencing. So if this is something that you're doing a lot of, PacBio has methods where you can take the output of many single cell platforms and make one of our PacBio libraries out of. And the benefit of this is you're going to understand the isoforms that are active in different cell types. Um, so it really is adding an additional level of resolution, even when you're doing single cell experiments. This is just kind of a table of, some of the use cases that people have kind of looked at this full-length RNA sequencing application and compared it to what they've seen with short reads, and they've, been able to show why this added level of resolution is um, important. Ooh, it looks like my PowerPoint crashed and is restarting, so apologies on that. I think we're back up. Um, So yeah, that's where, this is really, informative when you are looking at alternative splicing, trying to understand which isoforms are differentially expressed when you're looking at, discovery, or trying to understand, heterogeneity in cells. So if these are projects that, anyone in the audience is undergoing, this data type does have differentiated information for you to find. And I think one of the big questions we always have in this is, how many reads do I need for my project? And kind of, what we've seen thus far is, um, you know, the majority of sample types are typically going to, benefit from long read sequencing. So that's where we see, the average transcript length is somewhere between, this 1 to, 3.5 KB. We've also seen that at around 10 million reads, that's when we typically find about 80% of the known isoforms in a sample. So that's kind of where when we think of how many reads do you need for a project, it's kind of around 10 million reads a sample. That's kind of a good ballpark. And this is just, kind of a table of showing if you want to do, uh, quantification of this type of data, we typically look for around 10 million reads a sample, which is up to 6 samples on a Revio smart cell. If you're looking for discovery, you can go a little bit less. We're up, and up to 12 samples on a smart cell. And if you're looking for annotation, so if you're doing a lot of de novo genome work and you'd like to add annotation capabilities, that's where I'd say somewhere between 2 and 5 million reads or up to 24 tissue types on a smart cell kind of makes sense. And here are some of the kind of the recommendations of what we'd say you'd want to go into library preps with. So I guess, yeah, we'll move on to our final application space, microbial and metagenomics. And so that's where, on PacBio, we kind of see 4 flavors here. We see microbial isolate whole genome sequencing. We see, targeted sequencing, whether it's 16S or ITS regions. And then we see shotgun metagenomics, both in profiling and assembly. I think on the isolate sequencing front, PacBio is a great tool. It allows us to quickly, resolve and circularize contigs with one technology. When we look into these microbiome ones, PacBio, we can design amplicon workflows from the beginning all the way through the end. So we have protocols for 16S all the way from the wet lab through analysis. And that's similar for shotgun metagenomics as well. The benefit for PacBio and shotgun metagenomics is very similar to the RNA benefit I showed earlier, is with HiFi data, you're going to have longer accurate reads that allows you to both do assembly from metagenomics data and put these puzzle pieces together much more accurately than with short read data types. And this last slide on this section is just kind of showing from a microbiome sample. We were really able to fully circularize and pull out, many full bacterial samples from a metagenomics data set. So if there are any projects where you're looking to understand what is there, but that might not be culturable, this is a great technology to start to understand and look at these metagenomic communities in really high resolution. So that kind of concludes the last bit I have on applications. And so the last thing I'd like to share is just a little bit around project design and how to think about PacBio projects moving forward. Again, GIFs is the first Revio certified service provider in Canada. And it really is our high throughput, most cost-effective system. We recently launched the Spark chemistry, and there's really one thing I really want to point out with this. Compared to every other version of PacBio sequencing, this allows us to lower the input amount needed to make a library to put on the system. Previously, we'd require 1 to 2 micrograms of input material. With the Spark chemistry, we can get away with as low as 500 nanograms. So outside of being able to go in with less material, we also are getting about 33% more data per SMRT Cell compared to the previous chemistry. And so when we think of projects on PacBio, there are kind of two ways. One is based off of the insert size of your library, the amount of data that you'll get out. And the other can kind of be based off of application type. So that's where if you're doing microbial isolate genomes and you're okay with around 30x coverage, we'd recommend up to about 384 samples on a SMRT Cell. If you're looking at de novo genomes and you're looking at like a 3 gigabase genome, and you are okay with 20x coverage, that's where you could probably do about 2 of those on a SMRT Cell. Around here are a couple of other applications that may be of interest. So I guess with that, I'll go ahead and ask if there are kind of any questions.
Okay, just as Turner mentioned, thank you, Turner. As he mentioned, PacBio is supporting a sequencing grant opportunity to those that are attending today's seminar, providing a discount on Revio sequencing at GIFs. The winning projects will be awarded 50% off an RNA project and on 1 SMRT Cell. So don't do the one that needed 24 SMRT Cells that was on Turner's list of projects. We'll share the details on how to apply, and there's like a QR code and everything else. And as Turner mentioned, we'll take a couple questions, one from the room and one from online. Any questions for Turner in the room first? Any questions online? 2 questions online.
Hello. Okay, I do have one here. Can you please talk about the bioinformatics pipeline for human single-cell RNA sequencing using Kinnex data?
Yes, so I guess that might be one that it would be best for me to follow up with directly. So that's where what PacBio has is a couple of different tools. Namely, what I'd point you to is we have an ISO-seq tools data set on our GitHub page that kind of walks through everything from data you get off the instrument through deconcatenation and then individual steps of the process to get to what you're looking for with the single cell data. So that's something that you're happy to share directly as follow-up after this. But if you were to, just reference and look for our ISO-seq tools on our GitHub page, that would be a good place to start.
Great, thanks, Turner. We're going to switch to our next speaker because Dan told me I'm running too late.
Oh,
So Austin.
Time's on me too. Thank you guys, I appreciate it.
No, Turner, you gave good stuff. I took too long. Austin is up next. Austin Hammond, he is the platform lead for our sequencing lab. Austin joined GIFs about 3 years ago, and he's a graduate of the University of Victoria, not once, but twice, a bachelor's and a master's, and he trained in bioinformatics at the Michael Smith Labs at the University of British Columbia. And prior to joining GIFs, he was here at the Cancer Research Cluster at the University of Saskatchewan. So please join me in welcoming Austin to the front.
Great, thanks very much, Steve. Just not going to touch that. So I'm really excited to get to talk with all of you today about what we're able to do at GIFs at the high-throughput sequencing facility that we operate, and that I'm really proud to be a part of. What we have here at GIFs in Saskatoon is a truly state-of-the-art facility that lets us provide services across a number of really core disciplines to the biological sciences, specifically genomics, transcriptomics, genotyping, and bioinformatics. And one of the many distinguishing features of our facility is that we are a fully integrated facility that can handle sample types from a variety of sources, including microbial, plant, and animal samples, both tissues and extracted DNA. And our team of experts can support your project from its design through to its execution and data analysis, and especially providing that interpreted set of results that can be quite a sticking point in a lot of cases. A lot of providers will very easily give you raw results for you to then chew over for the next couple of years, and while that can be interesting and gratifying, quite often you need to move faster because time is of the essence. So we're happy to provide, help you to get to those insights. And one thing I want to do as I go through my talk today is highlight the individual members of the team that we have, because as Steve mentioned, the people that we have are so core to what we do, and it's their expertise that is essential for us to be able to provide the high quality results that we have. So here at one of our fleet of liquid handlers is Jacqueline. She's our automation specialist, and what she's able to get the liquid handlers to do is really quite remarkable, pushing the absolute limits of their capabilities. Some of the particular applications that we support are really almost anything you'd want to do with sequencing. But the key ones that I think a lot of people in the audience will be most interested in are our capabilities in the whole genome sequencing space. As Turner mentioned, HiFi sequencing really unlocks the ability to provide better reference genomes than ever before. But if you are already well familiar with what your genome is and you're at a later stage in your program, you may be looking for genotyping, and high throughput genotyping is something that we have a lot of experience with as well, for sample sets in the many thousands. Metagenomics is a key application. I'll talk about it a little bit more in a moment, but I want to highlight that on the RNA-seq side, we both support the Kinnex full-length transcriptome technique that Turner described so well as short-read techniques in the metatranscriptomics space as well, which has been very interesting to pair with metagenomics. And we are also able to work with submitted libraries from customers directly or in association with other partners to make available things like single-cell sequencing, which we have local partners able to work with us and provide those finished libraries for us to sequence, which we have an increasingly great amount of expertise working with. But the main thing we're all here to talk about, of course, is our long read capacity. Very proud to be the first certified service provider in this space in Canada, and that was a bit of a process to get through, but it was a collaborative one with PacBio, and it really centered on demonstrating to them that we can robustly and reliably produce high quality data. And they gave us the thumbs up, and that's why we're all here today. But long reads aren't the only technology that we offer. We also have two short read platforms that really excel in particular applications. Our newest one is the Element Aviti sequencer, which is a low to mid capacity sequencer, but its distinguishing feature is that we can use it to generate really highly accurate data that is, in some cases, tenfold more accurate even than Illumina reads. And just below the instrument there, we've got a shot of some of the team after we completed our install training for the Aviti just a month or two ago. From left to right, we have Sarah Yu, who you'll see in the audience here, running a microphone over to you if you have a question. On my other side is Sarah M., Marin, and Nikki, I think all of whom are here in the room today, so don't hesitate to introduce yourself afterwards and ask them your most challenging questions. Don't bring them to me. But if you have a very large project or need a huge amount of data on the short read front, that's when we move to our really high capacity short read sequencing instrument from Illumina, the NovaSeq 6000. On an Aviti run, we can do about 2 billion read pairs. We can do 20 in a run on the NovaSeq. So it is really the instrument of choice for those really high throughput sequencing-based genotyping techniques. Now, we're not going to ask the people on the team to make thousands of libraries by hand. While they would be fabulous, it would just take a very long time to do. So as Steve mentioned, we've been investing in a lot of the automated liquid handling instruments, and our fleet has continued to grow. We use them to produce a variety of library types, but as mentioned, we are the only service provider in Canada with the PacBio SmartBell workflow fully automated. And that has been a really important feature to support some of the large pan-genome projects that we have to turn around in a timely fashion. Now, these libraries that we make are supported by a really extensive QA and QC procedure, set of procedures. A couple of the instruments are highlighted here, but the one I really want to describe is our femtopulse instrument, which is a pulsed field gel electrophoresis piece of equipment that is the best way to resolve these high molecular weight DNA smears and get the really accurate sizing information, because of course we want to put in the best quality DNA and the longest DNA that we can for fragmenting to PacBio input to get those longer read lengths if possible and maximize the data that you'll get out of each of these smart cells. And one of these microbial applications that has come on the market very recently, and I believe we're the only provider in Canada offering this right now, is Longplex from Sequel. And it is really ideal for these smaller genomes because it generates fragment lengths that are best suited to those in the 7 to 10 kilobase pair range. But its main claim to fame, so to speak, is that you barcode the DNA itself prior to pooling it and constructing one PacBio library. That leads to substantial cost savings. It won't reduce the amount of data that you need to generate because those observations still need to be made, but it will let you be more efficient with your funds, putting those libraries together and getting them ready for the sequencer. And here we've got Gord, very focused on handling some liquids. He actually produced some really nice long-plex libraries very recently. Now, I want to take a few moments here to talk about our data management and analytics team at GIFs. We, as the sequencing facility, are very tightly coupled with this team because they are essential to actually getting data out to you, but also providing that interpreted data. So when we're generating billions of read pairs, they manage to streamline the data acquisition so that it happens in a timely fashion and securely. They are responsible and have constructed some really great QC tools for us to evaluate the data that comes off, ensure it meets the high standards that we have. They're also our bioinformatics team, so they are able to analyze your data either to a basic extent or quite extensively, depending on what your project requires, and then produce reports that are actually easy to follow and understand, and they look quite pretty as well. One of the analyses that is quite similar to something that Turner actually showed is shown here, which is looking at sequence similarity in two sets of chromosomes, and the overlapping ribbons are the ones where you can see similarity between different chromosomes in the two organisms. So that's, of course, a very sharp image, but it's also interactive, which lets you explore your data quite fully. 4 of the other specialties that I'd like to highlight are their expertise in genome assembly and annotation, especially these polyploid genomes can be quite complex to work with, but by leveraging HiFi data, we are in many cases able to even produce a haplotype resolved assembly, and that can be really exciting. Differential gene expression, a lot of us are used to that kind of quantification from short-read RNA-seq, but they also have expertise in the Kinnex full-length RNA-seq technique that we've heard already about. Variant calling, able to perform that key genotyping that is of great interest to a lot of members of the community, but they're also able to go further and look at certain structural variants, which HiFi is much better able to resolve than other short-read techniques. And finally, on the microbial side, microbial community classification and identification to understand, for example, what is the community of microbes present in some of your key soil samples. But now more than ever, these days, it's important that your data is secure and can be reliably accessed. So what we have is an isolated and secured network, and some of the key features that relate to its security are our regular audits, that we have remote data backup and disaster recovery plans, should they arise, but also physical access to these compute infrastructure. It's important to restrict from unwanted actors. So there's 24/7 monitoring by security officers, and in the event of a power outage, which I think we're pretty resilient to here in Saskatchewan, but when they do happen, we do have on-site emergency generators. And of course, your data is segregated and password protected. But despite all that security, it's still easy to access for you through our secure portals. And you're also able to download your data, if you're comfortable with it, from a Unix command line utility. Just as I get towards the end of my talk, I want to highlight 2 case studies that we were really excited to be able to deliver on for our partners. One was from a plant breeding company where they came to us with a very time-sensitive question. They needed high resolution, high quality sequence information for some of their elite lines so that they could make the necessary selections and continue on the timeline for their breeding program. So they delivered a couple dozen DNA samples to us from these key lines. We were able to construct the libraries, get the sequencing done, and make it available to them within 2 weeks of their delivery, which kept to their deadline. So they were very thankful, and we were really happy to be able to do that. At the time, it was the fastest we turned something around. I think we can go even a little faster now. The second project is a really interesting and ongoing livestock pangenome project, where as part of a consortium, they need reliable annual sampling from a few dozen individuals. And this was a really interesting case because we were able to accept the semen samples, perform the extractions, obtain high quality DNA, and then generate, of course, very high quality read data for them that met the requirements of their consortia. I think they were one of the few in that group that were using PacBio instead of the older nanopore technique, and it met and exceeded all the requirements they needed, of course. So that was a really nice case study to deliver to them and has formed an ongoing relationship, which is really gratifying. So what are some of the advantages of working with us here at GIFs? Well, as I think I've described, we have a team of experts who you may be able to find in the audience that can provide advice for your project's design and services along the span of what that project would encompass, from extractions to preparation, sequencing, and data analysis. And we are very collaborative and consultative to understand what the precise needs of your project are, and are really happy to talk to you. And if you have a large project, that is no problem at all for us to handle. We're comfortable processing thousands and thousands of samples, especially on tight timelines, really as a consequence of our investment in high throughput automated liquid handling. And for those very large projects, we are able to offer you volume-based discounts. So how can you work with us? As Steve mentioned, we've got the partner with us at gifs.ca address. That is an excellent way to get in touch with us, and then we can connect you to the expert best suited to help you get your project going with us. And we love acting as a connector and catalyst in the sector. So even if you have a project of a variety that we've never done before, we are part of this network, this really special network that we have in Saskatoon and globally. So we'll be able to connect, and I'm sure get a set of connections together to make what you need happen. And with that, I'll just leave up some of the information about the grant that we're running with PacBio up, while I'm happy to take any questions that anyone has.
Okay, thanks, Austin. It is no questions in the room, no questions online. We're going to go right to Andy because it's 11 o'clock and Dan is standing up and telling me he's given me the signal. Dr. Andy Sharpe is our keynote speaker today. Now, Andy got his PhD at the University of East Anglica in the UK. I've known Andy since 1997 when he moved to Canada. He was working at the time at Agriculture and Agri-Food Canada. And then Andy left Ag Canada and went to NRC, and I worked with Andy through a partnership with Dow AgriSciences and the NRC PBI at the time with Andy, was a key researcher there. And then lo and behold, I showed up at GIFs and Andy was at GIFs. So it's almost like our careers have, we've been joined at the hip. We're not twins, as you could tell, but Andy and I have had the opportunity to cross paths for a long, long, long time. Andy is a senior research scientist at the Global Institute for Food Security, and he and his team are leading lots of exciting discovery research projects, creating resources to advance plant breeding and plant discovery, and a lot of stuff which what like Turner and Austin had talked about. Lots of things that are relevant for us here in Canada: canola, wheat, camelina, many brassica species. And, you know, in 2023, he was part of a team that sequenced jackfruit. So when you go to the grocery store and you see this huge thing that looks gnarly, you know, in the fruit and vegetable aisle, it's jackfruit. And Andy and his team of partners were able to sequence that. And that's an important staple in many parts of the world. Please join me in welcoming Andy, or pardon me, it says Andrew, to the stage here.
Okay, thanks, Steve, for the kind introduction. I guess senior research scientist. Senior has multiple meanings, and I think each day that goes past, I'm getting closer to one particular meaning. But it's great to be here. Thanks very much for the opportunity to speak and update you on some of the forage crop research projects we have ongoing at GIFs. And thanks also to PacBio for supporting this event. Okay. So, yeah, the forage crops on the Canadian prairies, they're important crops for livestock production, of course, critical, of course. And there's actually a diverse range of options available in terms of different crops, which enhances the resiliency of pasture systems on the prairies. And there's actually integration of different legume and grass species, and collectively that improves the soil nutrient levels and ultimately the forage quality for the animals. There's basically you can select appropriate combination of the different species dependent on the particular area you are in Western Canada, which is kind of crucial for managing the different crops for the different livestock animals. And of course, the crops themselves are perennial, and you can have mixed pastures, and that's actually quite a nice habitat for wildlife. So it has an ecosystem impact as well, sustainability impact. I work very closely with Dr. Bill Legetu in these projects. He's the forage crop breeder at the Crop Development Centre at the University of Saskatchewan. That's him in the field in one of his trial plots. And yeah, I've been working with him for about the last 6 years, I think, in total. And of course, forage crops are an enormous role in Canadian agriculture. Collectively, about over $5 billion economic impact, about the third largest after canola and wheat. But you have a lot of species that make up forage crops, of course, as I mentioned, grasses and legumes. And we're working on 5 different species, and we'll be talking about 3 species in this talk: brown grass, hybrid wheatgrass, and alfalfa. And Bill's program, it's striving to improve the genetics, of course, of these crops, and make sure that the varieties that are released are adapted to Western Canadian conditions. And it has a breeding focus, covering a range of different agronomic traits. But some key ones are stress tolerance, in particular abiotic stress tolerance, to ensure the crops can thrive on marginal lands and drier environments. So brome grass, I want to talk about brome grass first. It's important both to pasture level and as hay. So you have a year-round impact in terms of forage for the animals, in particular cattle like this particular crop. It's very palatable and nutritious, providing good levels of protein and low fiber in the diet. It also has early growth in the season, so you'll get good growth this time of year, actually. and you have high yields. And they have good regrowth aspects as well. There's two types, primarily smooth brome grass and meadow brome grass. And it actually comprises some multiple species, but the ones that are commercially grown is Bromus inermis and Bromus riparius. And these are just two examples here. They look very close, of course. So only when you actually look at them in detail, you can see different morphological differences, particularly at the seed head level. The thing about this particular crop is that there's complexity, both in terms of the different types of species, but also genetically, there's big variations in ploidy. So you have diploids, tetraploids, hexaploids, octoploids, and decaploids. So you have significant variation in genome sizes. And as I mentioned, there's quite a range of different species. And because of these high ploidy levels, you have complex genetics. What that means is that you'll end up with, rather than chromosomes pairing with their homologue in the particular genome, in the polyploid context, there's that chance of them combining with other homologous chromosomes, so other genomes. So they end up being a bit promiscuous. So you can end up with an impact in terms of the genetics at a breeding program level. And of course, it's a longer breeding cycle as well. So the objectives of this project in particular is developing these foundational high-quality reference genomes at a diploid level, and then using them to characterize diverse germplasm. And then, based upon that data, also obtaining phenotypic data so we can identify markers that closely associate with genes or loci in the genome that have a beneficial impact on key traits. Biomass, plant height, regrowth, and protein and fiber are key aspects. So the approach, of course, we have to collect germplasm, and then we have to characterize that germplasm phenotypically and genetically. First of all, we wanted to identify diploid genomes, so we actually went through a process of doing that using flow cytometry and chromosome counting. Based upon that, we had target diploid genomes that we could target for sequencing. And then once we have the sequence, we can then characterize the rest of the germplasm at a molecular level using a genotyping by sequencing approach. And that ultimately yields markers that we can associate with traits of interest. This is just an example of flow cytometry. On the left is a plot of one of the target diploid genomes. And what you actually do is run the sample along with the standard genome. In this case, it's canola, which is a similar size. So you actually you're running it with a reference effectively. So you can actually get a good estimate of the target genome. So here we've got an estimate of 2.3 gigabase genome at a diploid level. And then we can actually confirm that by doing chromosome counting, where you can actually look to ensure that you've got the appropriate number of chromosomes in the accession that you're looking at. And here we see 2n equals 14, so we have two pairs each, 7 chromosomes, 7 being the basal number for a lot of grass species. We can also do KMA analysis, so this is where you're using raw sequence data to get an estimate of the genome size. So here we have two targets. These are the two target genomes that we went ahead for sequencing. We have flow cytometry analysis, chromosome counting, and then the KMA analysis. And this enables you to get an estimate of genome size based upon available sequence data. So it's whole genome, relatively high depth data, and this enables us to confirm in one case that we had about a 2.5 gigabase genome, and then the other genome was a bit larger, about 4.5 gigabases. Now, the sequencing, which is a key interest in this audience, is PacBio, of course. We actually had a sequel before we had the Revio, and you can see that just on the left of the picture there, which is now gone. The Revio is supplanted the older platform. So we had about 70-fold coverage of data, which is a bit higher than what you normally target, and that's because of heterozygosity, and I'll get to that in a while. And then also a lot of Hi-C data, so the chromosome conformation contact data. So 3C technology, but when it's in a sequencing context, we call it Hi-C. And then a software package, a pipeline using HiFiism, Hi-C Pro, and End Hi-C, and using those packages on the high-performance computing network that we have at GIFs, the University of Saskatchewan Copernicus, and managed by the Data Management Analytics group at GIFs. So the HiFiism gives you the base assembly, and then the other two softwares allow you to put together those basal pieces of the jigsaw into entire chromosomes. And then we also generated transcriptome data, both RNA-seq data, short-read RNA-seq data, and then long-read isoseq, again on the PacBio platform. And these are the assemblies that we generated for both Bromus riparius and Bromus sclerosus. So those are the two diploid genomes we targeted. And what we got was a total assembly size that actually aligned pretty well with our estimates from the flow cytometry and KMA analysis. And the nice thing here is that we got very large N50 contig sizes, so approaching 300 megabases in the case of riparius. What that means is that 50% of the genome is in pieces that are 300 megabases or larger. So essentially, you're getting to the point where you're getting close to whole chromosomes being assembled. On the right, we have the two genomes represented in a circus plot format. We have representation of genes, gene expression, and repeat content, and on those different circles within the plots. And on the bottom, we actually have the Hi-C contact maps. And what you want to see here is that bright red diagonal across the plot. What that means is that the Hi-C, what it is essentially its sequence points along a chromosome. And what you want to see is those points essentially joining up the contigs that you have, or confirming the assembly quality that has been generated in the base assembly. So if you start to see patterns that go off and look and actually matching other parts of other assembled contigs or chromosomes, then you know you've got an issue. So if you see a nice diagonal, it means you've got a faithful representation of the genome. Of course, I mentioned the heterozygosity. So what that means is essentially you've got two genomes in one. So these are outcrossing species. Actually, all the forage crops that we're looking at are outcrossers. So what that means is that you, rather than having each chromosome pair being identical, there is actually differences at a single nucleotide level and also at a structural variation level. And that's actually represented here in this plot, where we're looking at the two haplotypes from the riparius genome across the 7 chromosomes. So you can actually see there's some differences here. It's you probably need a close-up, but yes, we've got essentially a collinearity plot between the haplotypes. And we see there's, in red, we've got some inversion points. So we've actually identified bits of the genome that's inverted between the two haplotypes, and that's confirmed with the Hi-C data for the scaffolding. And then we actually see some duplication of genes between the two haplotypes as well. So essentially, you've got two genomes. I won't say exactly for the price of one, because you have to get more PacBio depth, but it's quite nice when you see the quality of these outputs. And we see variation in repeat content between the two species. So riparius and sclerosus are represented here. So the black bits in the center represent the centromeric regions. And we know that based upon the repeat content. So there's actually layers of different repeats above each of the chromosome bars. In red, we've got a centromeric repeat. So we see that obviously enriched in the two genomes at the centromere region. But you can see there's a lot more in the sclerosus genome compared to riparius. And overall, there's a significant difference in repeat content between these two bromous diploid genomes. So looking at a bit of a close-up here, comparing chromosome 1, this is to scale. So you can see there's a larger chromosome 1 in Squirrus compared with Riparius. And you see it's a larger repeat content. So it's expanded repeats in Squirrus, which is making the difference. But overall, the synteny, the collinearity between the two is at a genic level is pretty close. There's some differences, but overall you get the same order of genes along there. The chromosomes is just the repeats between the genes which are different. And then looking at the repeats themselves, we can actually assess when these expansion of repeats happened in the two genomes. So we can do this analysis where essentially it's a molecular evolution analysis, and we can tell, get an estimate of when the expansion events occurred. And we see that in Riparius, in the blue, then you're actually seeing more recent expansion of repeats, whereas sclerosis is actually expansion events older compared with Riparius. This is based upon full-length LTRs that have been identified in the two genomes. Again, that's something you get represented quite nicely with these PacBio long-read assemblers. We can compare the genomes. So we have Riparius and sclerosis represented in this dendrogram plot. And then we have two other bromous genomes that were published in the last 2 years, Tectorum and Sterilis. And obviously we see those 4 genomes clustering together, they're related. And on the synteny plot next to that, we can see that overall the order is fairly similar, with some events separating the two genomes. But there's actually an evolutionary conserved difference, translocation between chromosome 2 and 5 when you compare these genomes against one of the next closest relatives, which is barley, the H genome represented by Holden and Vulgari here. Looking at the genetic diversity, then we had several hundred accessions, both from the Plant Gene Resource Centre here in Saskatoon at Agriculture and Agri-Food Canada, and also from the USDA collection. So basically looking at about 15 species, various numbers of accessions in the different species, and from all over the world, primarily perennials, but also some annual species as well. We've grown out these different accessions, both in the greenhouse context, and that allowed us to collect tissue for DNA extractions, but also we can carry out some morphological analysis. And then we've also grown out these accessions in the field where we essentially you can clone these plants that you get fairly effectively and get multiple plants that we then transplanted to the field. And we also did flow cytometry here. So you can see the range of different genome content here, diploid, tetraploid, hexaploid, octoploid. Population structure and analysis, which allows us to work out the number of different populations amongst this collection. You can see that primarily we've got big groups for meadow and smooth brown grass, represented by green and light blue here. The dark blue on the left is actually a collection of double haploid lines, which are generated with collaboration between Bill and the NRC, and we expect those to be looking pretty similar. That's confirmed with this dendrogram analysis. See that the higher divergence among the wild relatives on the left-hand side, the centre on the left-hand side, is quite significant compared with some of the cultivated brown grass species, so Bibastini, inermis. And there's some breeding lines, and then those DH lines are all clustering on that right-hand side. The morphological characterization we're using was in the greenhouse, but also using the Trait Seeker, which is our digital phenotyping platform, which is running, I guess last year it was running at the Livestock and Forage Crop Research Excellence Centre out at Clavette. And so we have these plots, perennial plots, so each year we're getting the plants growing up, and we can carry out these analyses, so we can capture data manually by eye, but also digitally with this platform where we have various sensors that detect at different wavelengths. We can then actually collect information about plant height, biomass, vegetative indices, and then there's actually a LiDAR system in there, so you get a 3D picture of the canopies. So it gives us greater accuracy for monitoring different aspects, including plant height. So that's what we're doing in brown grass. I'm going to quickly move on to alfalfa, which is the legume crop, again, outcrossing, a key high-quality protein source for livestock. Of course, key also for soil productivity because it fixes nitrogen. But there's some limitations in productivity because of stresses on this crop, in particular drought stress and salt stress. So there's actually a significant yield penalty in this crop when grown under drought and salt stress conditions. So that's part of the one of the key objectives in this project is assessing at a phenotypic level salt and drought tolerance, and then looking to see if we can associate genetic variation with loci in the genome or genes in the genome that have beneficial impact on these two traits. I'm not going to go into that. I'll just mention the reference genome creation, again using PacBio, and using a similar pipeline to what we used for brown grass, a base assembly with HiFis and Hi-C data, and then ultimately we do gene annotation and repeat annotation for the final assembly. For the diploid, then I'm just going to present the diploid genome that we have. This is an ongoing project, so we're still generating outputs for it. Again, 2 haplotypes represented because it's highly heterozygous. But we've got, again, nice scaffolds and contigs generated from the base assembly, and essentially looking at easily being able to generate chromosome level assembly. And again, we have the Hi-C contact map. We see that nice diagonal line across the plot, meaning that we've got a faithful representation of the 2 haplotypes in this diploid genome. Hybrid wheatgrass is another crop, again, another grass. This actually has some significant tolerance for drought and salinity, but Bill would like to see more. So that's part of the effort that we have in this project is, again, to identify variation that will increase that tolerance. There's actually an impact here in bread wheat because one of the parental ancestors of hybrid wheatgrass has significant disease-resistant loci. So there's been introgression into wheat of one of the parental ancestors. So the sequencing effort that we would like to result in is faithful representation of those ancestral genomes. The hybrid wheatgrass genome itself is hexaploid in nature. It's allohexaploid. So we've got multiple genomes, H genome and 2 copies of the S genome. It behaves as an allohexaploid, so the assumption is that there are 2 different types of S genomes. But we wanted to confirm that in the assembly. So again, similar pipeline, HiFi assembly, Hi-C data, and we get a final assembly produced. For this flow cytometry analysis, you have to use different standards for the different size genomes. So for this one, we use wheat and durum wheat as control or as reference standards. So it enabled us to give us a size which we thought was between 11 and 12 gigabase genome for hybrid wheatgrass. The assembly, using about 10 flow cells of PacBio data, about 700 gigabases of data, HiFi assembly, Hi-C scaffolding. Again, we get 2 haplotypes. It's heterozygous, but we ended up again with nice plots represented, the nice diagonal. You see that, there's no pointer, but there's some shadow diagonal regions on these plots. What that actually is reflecting is the polyploid nature. There's actually relatedness between the different chromosomes in the different genomes. So you see a bit of contact association. That probably is because those 2 genomes are most closely orientated in the nucleus. So when you actually extract the nuclei and extract those contact points, the homologous genomes are closely associated. So you see this crossover effect. We can't really see this very well, but what we did is do a comparison of this new genome assembly, because we didn't know which were H genome, which were the ST1, and which was the ST2 genome. So what we did is compare it with a recent assembly that came out last year, Elymus cibricus, and that allowed us to identify the H genome fairly effectively. On the dot plot on the left, there's some faint bands. And what that's actually reflecting is homology between our assembled chromosome and the assembled chromosome in the paper. So that actually allowed us to identify one of the H genome and one of the ST genomes that are homologous to the cibricus, which is a tetraploid. And then once we actually did, you cannot see this at all really, but we actually did a lower stringency dot plot here to identify that we actually asked the other scaffolds where we didn't see homology, we actually did get some homology with that of the genome. So basically what this is confirming is that we've got two quite different ST genomes in hybrid wheatgrass. As expected, it's a allo hexaploid. Okay, summary. So we're developing new high quality annotated reference genomes in these different forage crops, using PacBio, HiFi and HiC data, brown grass, alfalfa, hybrid wheatgrass. So these assembled genomes allow us to do comparative analysis with related genomes, including well-characterized model genomes. That's kind of key because there's obviously been a lot of research in those model genomes, but there's some close similarities. So that's important when you want to translate information from model genomes to the crop. You see utility of assembled genomes for diversity analysis and genetic markers discovery. So that ultimately allows us, we can use the data to carry out trait association analysis, ultimately have markers that can be used in the breeding programs. This year we'll be starting two new projects, Sisa milkvetch, another legume species. That actually is important because it provides bloat resistance that you see with alfalfa. That's a common issue with alfalfa. And then Timothy grass. It's another grass species, a common forage crop out in eastern Canada, but also in the Peace River region of Alberta. Finally, acknowledgements. Of course, I have to thank Bill and his partnership. He's been great to work with over the last half decade. And then Byamba in his lab is kind of key for organizing the field work for us. And then Raju for brown grass, Irfan for alfalfa, Nadeem, and Yan for hybrid wheatgrass. They're the key players. So some of them are in the room at the moment, who played a key role in leading the technical aspects of these projects. And then Ling Ling, she's the lead supervisor for Yan. I'm the co-supervisor for Yan. But she provides great input on the analysis side. Sampath, who was played a role in the technical management of these projects. Kevin is a key, he's the lead for DMA, and the DMA team is actually critical for enabling us to do this work and the visualization tools. And Zhang Peng has provided great technical support for a lot of these efforts. And of course, the Opal team at GIFs is key in producing the data. And of course, our funders. I guess the Government of Saskatchewan provides the bulk of the funding for these projects, but we also get co-funding from Saskatchewan Cattlemen's Association and then the Saskatchewan Forage Seed and Development Commission. With that, I'd like to end, and I'll take some questions if there's any time. But
yeah. I got to lower this because Andy's a bit taller than I am. So again, just a reminder for questions in the room, put up your hand and we'll run a mic over to you. And for those online, please type your questions into the chat. And Scott, you did not disappoint me by raising your hand first. Thank you, sir.
Thank you. Is there an effort to look at the success in markers and actually move them out into the field? And so you've got marker identification. If you're actually doing now a breeding program for specific markers, call it ADF, right? And analyzing the effects. So in essence, kind of moving to a EPD basically.
Yes, Can you hear me? Is that, yeah. Yeah, that's a key aspect, is moving, translating this discovery into the field. So we can do that through the breeding program. So we identify these, essentially it's variations in the genome, which are associated with beneficial phenotypic aspects. And it depends on the particular trait you're interested in, but you end up with various regions across the genome, loci across the genome that have a beneficial impact. What we ultimately want to do is actually assess all of those simultaneously in your breeding program. So we have a panel of markers which we can use in the breeding program. And what that will enable you to do is ultimately pyramid good genomic combinations together. So you actually can put everything together. So you basically can do this using a essentially called a genomic selection approach. It's a whole genome approach that's incorporated into the breeding program. And that's what we want to build to in these research projects, the outputs from these research projects.
And so is there a confidence interval, if you will, for the team phenotypic characteristic versus the genomic?
Yeah, that's right. So we run stats, of course. The nice thing is that nothing working with plants is that you can generate nice populations and grow out right, and you can structure the population. So genetically, even though the genetics are complex because you've got multiple genomes, with the polyploidy, you end up with a structure that allows you to get really high confidence statistical associations, which you can have faith in working in the future in the breeding program. Yeah.
Any other questions? So Scott's the director of the Livestock and Forage Center of Excellence, where Andy and team were driving the phenomobile down. So thank you, Scott. Anything else in the room? Okay, Andy, thank you. Austin, thank you. Turner, thank you for all of you for presenting. At GIFs, we take pride in being a connector, and it's events like this that allow us to bring different people together to get things talked about. And usually ideas, there's people in the room I know that have ideas that are too afraid to say it, but that's okay. They percolate, connect with us, eager to help you advance your ideas. And again, reach out to us through Partner With Us at GIFs. And I also want to remind you of the PacBio sequencing grant that's running, and you can see the QR code with the Tyrannosaurus Rex on there. The recipients will be awarded 50% off an RNA project and one SMRT cell, and you can apply using the code. And for those in Saskatoon, you're welcome to join us for a lunch. But before we do, I want to invite